My talk is going to be quite a personal history of where I think my laboratory has made some contributions to the field of nanomedicine. I'd like to introduce you to Dr. Alec D. Bangham, if you haven't heard of him already. This is a portrait of him in his house with his liposomes up here in the background. He was the first to describe multilamellar smetic mesophases, as he called them, which was termed Bangasomes, but now we call them liposomes or lipid nanoparticles. He proposed that they could be used to study the functions of cell membranes and also as an artificial lung surfactant in premature babies. So that was the first commercial application for them. But before long, researchers such as Gregory Grigoriadis at University College London and Dimitri Papadopoulos at the University of California, San Francisco, and some of our colleagues were proposing that liposomes could be used for drug and vaccine delivery. Early on in my laboratory, we actually used them to study functions of cell membranes. So we use them to study membrane fusion and to reconstitute membrane proteins. And at the bottom, you'll notice that I put, I think, what are some of the key references. I want to show you this picture. It's a picture that was taken in Berlin in 2002. With myself and Dan Lassick, who was with Liposome Technology Incorporated, Alec Bangham himself, and Dimitri Papadopoulos. And this picture is notable for the fact that I'm the only one that's left alive of these three, of this group of three pioneers in the liposome field. So I rather treasure this particular photo and consider myself lucky that I've survived. In the early days, drug carriers had two major challenges that limited their clinical applications. First, they were rapidly removed into the reticuloendothelial system. And in 1987, we published the first paper that showed a way to keep liposomes for circulating longer in the body. And we initially used the monocyalloprotein GD1 to show that. And then we did quite a bit of research on the use of polyethylene glycol, which we actually now term PEG. The other problem was that drug loading was inefficient and drug leakage rates could be very high from the particles. So Doctors Deemer, Barinholtz, Cullis, and others pointed the way to efficient drug loading systems and ways that the drug leakage could be reduced from the liposomes, particularly for weak base anti-cancer drugs. And this research together led to the product Doxil, pegylated liposomal doxorubicin, which received FDA approval in 1995. And currently, the worldwide sales of Doxil are $1.25 approximately $1 million. So it's one of the poster childs of drug delivery using liposome. And there is a couple of my early publication on the long circulating liposomes containing GM1 and also containing PEG. The next challenge was demonstrating proof of concept in animal models, and then following that, finding ways to increase the selective delivery of liposomal drugs to target organs. So we demonstrated initially in an early paper in 1992 that a sustained release drug delivery system of cytarabine, soluble anti-leukemic drug, significantly reduced the toxicity and increased the lifespan in an animal model of leukemia compared to the free drug. So that was an early proof of principle for animal model applications. And then we also subsequently developed and tested a number of ligand-targeted liposomal drug delivery systems and published the first demonstration of their increased efficacy in vivo compared to non-targeted systems, where the liposomes were targeted using internalizing ligands. So something like an antibody or another type of ligand that attached to the surface of the liposomes and could trigger the liposomes being taken into the cell, so that many of these drugs that act intracellularly would be able to get into the cell and have their actions. And I've actually, I'll just bring your attention to one paper down here from 2002 that shows that internalizing antibodies are actually necessary for the improved therapeutic efficacy of antibody-targeted liposomal drugs. In other words, the liposomes have to get inside the cell and release their drug internally. We also became interested in gene therapy quite early on. Phil Feldner had published, and of course, he'll be talking also in this symposium. He had published a number of really exciting papers demonstrating the efficacy of cationic lipid systems for transfecting cells in vitro. And we were looking for ways of increasing the efficacy of cationic liposomes relative to the gold standard of that time, which were retroviral vectors. And we wanted to deliver DNA into cells. And so over 25 years ago now, we showed that asialofetuin targeted liposomes increase the expression of a beta-galactosidase reporter gene in hepatoma cells. So that was one of our early forays into gene therapy. A lot of liposomal drugs are used for chemotherapy, and chemotherapy as it's practiced in the clinic depends on combinations of drugs with non-overlapping toxicities and side effects, non-overlapping side effects, that is. So we started looking at liposomes containing combinations of drugs and combinations of targeting agents. And so we got into trying to mimic the chemotherapy by using combinations of drugs and combinations of targeting agents. And so we were looking into a lot of models where we, like B-cell lymphomas, as you see down here, where we use combinations of doxorubicin and vincristin. We use targeting combinations of targeting agents like anti-CD20 with anti-CD19 and anti-CD22. We targeted via whole antibodies, Fab fragments and scFv fragments to see which were going to be best. And I have to say that out of those, we actually, not from my laboratory, but the first liposomal combined drug, Vexus, which contained the combination of daunorubicin and cytarabine, was approved by the FDA in 2007, and so it was reduced to commercial practice. Now, controlling the surface properties of liposomes was not only time-consuming, but difficult to scale up. So we developed methodologies for changing the surface properties of preformed liposomal drugs. So Doxil and other liposomal drugs were on the market by now. And so we were looking for ways to convert them into targeted agents. We wanted to find a technology where we could easily convert the commercial liposomal drugs to have improved properties by just inserting some ligands or antibodies or polyethylene glycol into the preformed liposomes. And so that was actually quite successful. And I have noticed that the post-insertion technology is actually being used more and more these days as a simple way of converting an already commercial liposome into having other properties like targeting ability. We also spent a lot of time doing research into the pharmacokinetics of liposomes. Now, the circulation half-life of liposomes and their biodistribution in vivo is pretty critical to their therapeutic effects and their side effects. And we performed many studies to examine the effects of size and charge and dose and route of administration and the effect of surface modifications on the pharmacokinetics of liposomes. And developed methods also to measure the degree of drug bioavailability in target cell populations like tumor cells in the body as a function of the liposome properties. And ultimately, we found that pegylated small unilamellar liposomes had a bit of a sweet spot in terms of their long circulation times over a broad dose and a broad range of sizes. And of course, Doxil fits right into that dose and size range. The other thing that we wanted to do was solve an important problem that came out of Phil Felkner's work, which was strategies to keep cationic liposomes circulating in vivo. For liposomal nucleic acids to show therapeutic effects in vivo, several important considerations have to be met. First of all, efficient entrapment of nucleic acids like mRNA in liposomes requires cationic lipids, but cationic lipids had problems with not only with increased toxicities in vivo, but also problems achieving long circulation half-lives. They ended up in the liver particularly avidly. And so we learned from our pharmacokinetic studies that positive charge can't be on the liposome surface. And we developed methods to make the surface of the liposome neutral with the positive charge and the nucleic acids trapped inside, which is really very similar to how they're making the mRNA vaccines now. And also with PEG on the surface to increase circulation times and inhibit aggregation. One thing that we did a little bit differently was that we wanted to increase the binding of the liposomes to the target cells when the target was reached. And that tended to be inhibited by PEG. And so what we did was we introduced specific internalizing targeting agents on the liposome surface to promote them to bind to the target cells and promote them to be internalized and release their contents within the target cell. And so originally we did this work and we patented it in 1998. And it was issued in 2000. And there's a reference here to where we use this method to produce a novel, long circulating and functional liposome formulation of antisense oligonucleotides targeted against MDR1 in cancer cells. So that was almost 25 years ago that we actually did that work. The patent, by the way, has now expired. We also looked for proof of principle in animal models of these targeted cationic liposomes. And we demonstrated one of the holy grails is to reach extrahepatic targets, such as solid tumors, with these cationic liposomes carrying nucleic acids. And we teamed up with a group at G. Gaslini Children's Hospital in Genoa, Italy, the group of Mirko Ponzoni. And we were able to show that cationic liposomal antisense oligonucleotide alone and given in combination with doxorubicin in a murine model of melanoma was able to increase the lifespan of the mice relative to all of the controls with and without targeting and so forth. And also a paper that I think was really one that I liked a lot, was that we along with the people in Mirko Ponzoni's group showed that cationic liposomal alkinase, siRNA, was able to increase lifespan considerably in a murine model of neuroblastoma. So basically, we got proof of principle of how to get extrahepatic targeting to solid tumors in animal models of these cationic liposomes, which is another one of those holy grails. Now, the holy grail is actually to get this happen in a human in vivo, but this sort of set the stage. So now, looking to the future, my research laboratory is now closed. I closed it actually about 10 years ago, but I continue to be active in administrative positions and in doing some consulting. In the meantime, Peter Cullis and Hesse Barinholtz, Lawrence Mayer, Tom Madden, and others have been instrumental in commercializing several liposomal nanoparticle products. But the COVID vaccines using lipidic nanoparticles as carriers of COVID mRNA are seen as the most successful of all lipid particles, with sales in the billions of dollars. So this really represents the maturation of our field and opens the door to a myriad of follow-on products to treat genetic disease, rare diseases, neglected diseases, and currently incurable diseases. Thank you for your attention. That got rather small. And I just wanted, at the bottom, I've actually put a reference to a website to a conference that I'm co-organizing in June on the use of nanoparticles in the treatment of a variety of diseases looking towards the future. So thank you for your attention.